All right, I think it's probably wise for us to go on and start the process and start this forum, because we do actually have, I'm very thankful for all the seminars, or for all the speakers and all the forum, and we have a very important topic, and it is something that we are all addressing with regard to our applications to NIH, and what the way we're going to handle this and the way we're going to run the presentation is very simple and very casual. I'm going to give you a quick overview of the issue we're facing with rigor and reproducibility and transparency, or as we call it, R2T, and just talk about its history, and then we'll allow each of our panelists to introduce themselves and give their background, as well as what their specialties are, and also the study sections that they have been reviewing for, because we try to get a wide range of study sections, because these criteria that NIH has placed out there vary across the study sections, and we wanted to see how each one is trying to address them. So as we begin, just a little bit of history. Going all the way back into Nature 2014, there was an article about NIH plans to enhance reproducibility. And the director, Francis Collin, actually wrote an article talking about some concerns about the self-correcting nature of publications, and the fact that a large amount, or a perceived large amount, of published literature has difficulty in being reproduced, and we have to work on a common definition of what we mean by reproduced. But at that time, NIH indicated that they were going to release 2 announcements emphasizing that there would be changes in the way all applications, the K, the Rs, the program projects, all of them would be reviewed. And there would be new emphasis placed on certain criteria, and 3 of these criteria would be scorable, and 1 of those criteria, if not addressed, though it's not scorable, could delay funding of an application. And specifically those 4 focus areas, and we've spent a lot of time over the past year developing a vast web library. I'll show you in a little bit of where these resources are. But the 4 criteria are that study sections are encouraged to discuss and score, along with the review of the application, is the scientific premise. Now, you may by default think the scientific premise is nothing more than the scientific literature review that we've been doing for years. Well, that is part of the truth, but that's not the complete truth. From my understanding of what NIH wants out of scientific premise, and based on what can be found online, they want to be able, for you to be able to criticize and critique the gaps in the literature, not just what others have done in demonstration of knowledge of what they have done, but where are the gaps, and how is your proposal and your methodology going to overcome those gaps and advance? And it's not just a criticism of other individuals' published literature, it's also a discussion of possible gaps and criticisms within your own preliminary research. How are you going to address those? The one that's drawn a lot of attention, we've gotten a lot of requests for help from, is with regard to the authentication of key biological and chemical resources. How are we supposed to do this? This is the one that is not scorable, actually. There is no score, and this is not going to affect your fundable or the score that you get from study section. However, you are required to upload a separate document, 1-page document indicating a plan if you are using cell lines or chemical reagents. How are you going to validate that they are detecting or producing what you have said that they will produce? The emphasis I've heard from study sections and some project officers is they do not want to see a one-and-done plan. What they're looking for is a strategy of throughout the process or throughout the grant. How are you going to be able to continually make certain that these things are working in the manner that you believe they should? This also, one of the things that could also be calibration of equipment. The relevant biological variables has mainly been focusing on really including sex as a biological variable in animal studies, but also it comes into understanding things such as age and accounting for all these within both clinical studies and animal studies. And then the one that's most dear and near to my heart is the rigorous experimental design and the statistical rigor that is supposed to go. Now, all of these to a certain degree have always been part of all our applications. However, now there's a greater focus, and they are supposed to emphasize these in the review. And with that, I'm going to start going around and letting the panelists introduce themselves. And again, the panelists to introduce themselves and give me background. We're going to start with Tulane, but just what is your background and with regard to your specialty and also the study section that you serve on. And I'll start with you, Dr. Burnell.
All right. Well, I appreciate the opportunity to join you today. So I'm Bruce Burnell. I'm the director for the Center for Stem Cell Research and Regenerative Medicine here at the School of Medicine at Tulane. We focus on using various stem cell populations and tissue engineering approaches to try and treat various diseases. I won't get into the specifics today. I think what's most pertinent for our discussion today is I sit on the TAG, or Therapeutic Approaches for Genetic Diseases study section. I've been on it for 3 years, 1 more year to go. I'm a free man. And I also review on a couple of panels that review SBIR and TTF grants, although I haven't done that yet under the new guidelines and new policies. The last time we did TAG was the first time we saw applications with some of the topics today. So it'll be interesting to hear what other people have experienced on their study sections.
Dr. Liu?
Hi, thank you, Dave, for doing this. And my research, I'm the professor of the Department of Biochemistry and Molecular Biology. I'm also acting chair for this department too for over the last 4 years. So my research is basically focused on the cancer mechanism, metabolism, and biology, and mostly involved in the most important tumor suppressor called P53. And my research book basically trying to understand how signaling pathway responds to different kinds of stress to activate the PP3, and also try to utilize this knowledge to develop some drug that potentially activate the PP3. So I have some drug discovery program developed over the last half a decade or so. And in addition to that, we try to use animal systems to understand biology that involve the regulator of the PP3. I think that's basically briefly summarize my research. In terms of my experience, I think since 2000, I've been serving a different NCI study section. The study section involving cancer, probably more than 5 or 6 different study sections. I used to serve as a Patho B as a permanent member. I don't know any of you here served at that study section before. Later on, that study section split into 2 different program study section, and now actually a standing member for DMP drug is called Drug Discovery and Molecular Pharmacology, and currently actually I'm reviewing the PO1 grant now. But basically that summarizes my background. Thank you.
Thank you. Vishnu?
Vishnu Subramaniam, Assistant Dean for Research and Graduate Programs at Auburn. A major research focus currently chemotherapy used in memory loss, chemobrain, also the diabetes Alzheimer's link. I'm a neurophysiologist, serving in study sections for the last few years, and recently had the chance to review some SBIR grant, where I noted how these criteria, what criteria has impacted the overall scores and how some applicants strategize or use strategies to address these concerns.
All right. Dr. Easley, Chris?
Yeah, Chris Easley, Auburn Chemistry and Biochemistry Department.
Our research is focused on using microfluidic methods to understand dynamics of hormone secretion and nutrient uptake dynamics from endocrine tissue related to diabetes and obesity and metabolic syndrome. We also develop all-volume immunoassays using acid immersion antibodies at the, say, picoliter to nanoliter scale on these devices. I've served for about the past 5 years on multiple panels, normally related to technology development, so the Enabling Bioanalytical Imaging and Technology panel, EBIT, the Instrument and Systems Development is the one that I served on this summer. In that one, we did have these new criteria involved there. And I've recently served on a panel. Just the second one I'm reviewing grants for now is a rapid assessment of the Zika virus, special emphasis panel. So we have really, I guess I've reviewed grants for the third time now under these new rules. And I think that will be it. Yeah.
All right. Dr. Gillespie.
God, what a picture. Thanks for picking that out, Dave.
You're welcome.
I'm Mark Gillespie. I'm professor and chair of pharmacology at South. It's a job I've been doing for way too long, over 20 years. My research interests are in transcriptional regulation, particularly in lung disease. We're taking a strong genomic bent now. We'd like to know why non-coding regulatory sequences are prone to mutation and how that affects transcriptional regulation. In terms of study section service, I've been chair of the lung biology and pathology study section, which morphed into a couple other study sections a few years ago. I had this target permanently tattooed on my back from all the study sections I've been on over the years, including standing study sections and SBIR and K study sections, and I'm now on this PEDAL protocol review panel for prevention and early treatment of acute lung injury. I've been on a few study sections, including a couple of P50 study sections and one regular study section in the last year since the R2T became activated.
All right. Thank you very, very much. And then the panelists that are here with us at UAB, Dr. Levitan.
My name is Emily Levitan, and I'm an associate professor in the Department of Epidemiology. My research is primarily related to cardiovascular disease and heart failure, though I'm involved in a lot of projects as sort of a methods person for population science kind of research. I've served on several different study sections as an ad hoc member, and most recently the Cancer, Heart, and Sleep Epidemiology Study Section, Panel A, and that is a fairly broad study section, all related to epidemiology.
Dr. Zhang.
So my name is Jianhua Zhang. I'm associate professor in UAB Department of Pathology. My research interest is on neurodegenerative diseases, Parkinson's disease in particular. We study protein toxicity and the clearance of toxic and damaged proteins and organelles by autophagy and lysosome pathways. I've served in several different study sections ad hoc, and the past cycle I served in the NOMD, neural oxidative damage something.
Okay. Well, thank you very, very much. And we're going to walk through, I'm going to leave this slide up for a little while, but we're going to go, as I'd already reached out to all the panelists and specifically set up kind of the order of things that we would discuss. And what we'll do first is we'll go through actually the authentication of the biological and chemical, key biological and chemical resources, mainly because this is the one that we've gotten a ton of questions on over and over with regard to, we get plenty of questions with regard to scientific premise and the other ones, but this one brings questions with regard to, what strategies can be put into place that convince the review panel that you thought this through. And I'm going to ask the panelists with regard to their experience for study sections without revealing the details of the specific grants. Were there specific strategies or approaches that seemed to resonate with the study sections to lead to a convincing belief that the applicants had thought this through and provided a clear strategy for authentication? And I'm going to, try to keep it organized. We're going to start, and again, if your study section really is not focusing on this, all you have to say is, well, we really didn't focus on that. But mainly I'll start with Tulane and Dr. Bunnell.
Sure, happy to. So just a couple things before I start into that specific topic. I'm not sure if this happened with everybody, but when we first started to get these grants, so the last round of review, the NIH people that run our committee and some of their ancillary folks had a pre-meeting conference call where they essentially did what we're doing today to some extent, except gave us the NIH's view on all these different topics. And the one thing that became pretty clear is they really don't know what to expect for many of these different topics when they see a grant application. So I think it's one of these things where we're both learning as we go. On to this specific topic, in our study section, again, TAG, this was focused on fairly heavily. Again, it's not really a scoring influence, but we did spend time on each grant that was discussed looking specifically at this. And I'll say, because this was the first time, the responses ran the gamut from people who completely ignored it and included nothing to people that were amazingly thorough. And I will say, you asked us to stress kind of what were some of the things that resonated with the review panel. Thoroughness clearly resonated. You could tell the people who really thought about this and put a lot of time and effort into it, basically just by looking at the plan. The other thing that really resonated with me and some of the other review members is, for example, if you're using a cell line and you're going to be using that throughout your study, repeat analysis of that cell line, showing that the characteristics haven't changed or the biology of that cell line hasn't changed, was really important. We wanted the PI to essentially say, I know these cells may not always be what we think they are, so I'm going to repeatedly look at it. We didn't really get into equipment in terms of calibration and all of that. People didn't really seem to think that was an issue. In terms of if it was a kit purchased from a reputable scientific company, we didn't even care if they addressed it because we assume that the company was going to sell a validated kit. So the things that really we wanted to see them stress were kind of homemade things, for lack of a better term. So if you were developing a cell line in your lab, or you were going to take a cell line and modify it, or you were going to do something else, build a plasmid, that type of stuff, we really wanted you to focus on the homemade things that you were going to generate in your studies or use in your studies repeatedly.
Okay. Dr. Liu?
Yes. I went to the last study section for the DMP, and believe it or not, most reviewer, even including the chair SRO, didn't really emphasize what this slide shows. And it was me to mention that, bring this out after a few round of review. First 4 or 5 R1, I didn't hear anyone actually mention all this legal transparency or reproducibility, none of them. So then I says, oh, was we supposed to comment on those things? And the panel kind of like joking around a little bit, and so didn't pay a lot of attention. But through all the grant that we review, and I think a lot of this applicant really address those issues, and others have not. But because the authentication is not really a score driving stuff, and I see one beautiful applicant who actually put a few pages right after the biosketch to authenticate anything that they use key reagent, cell line, plasmid, and animal model. So I know, to my knowledge, I personally, when I put those grants up, I have 1 or 2 pages, basically 1 page to describe how we authentify the key reagents. So there are a few companies, if you have animal model, Jackson Lab and Charles River, they can authenticate your animal model. If you have a cell line, then at least two companies can do that. I think where you got your cell can do those as well. So I think those, because these are not score driving components, And if a grant missing this, they may not make a big deal of it, but they will recommend you to do that. And not only that, because when recently we have a few papers accepted by or reviewed by journals such as eLife, Nature Communication, Nature Journal, they actually ask you to show the authentication of your reagent, like cell line. And some of the reviewers really put the comment on this. I just want you to pay attention to that. That's my personal experience to share with you.
Okay, thank you. Vishnu?
Yeah, it's pretty much what the first panelist said. Some applicants addressed very nicely the authentication, and the SRO spent about 15-20 minutes discussing this prior to the meeting. They also said that it has to be less than 1 page, and authentication data is not required. In fact, they don't even ask you. It's not required, and they don't want you to provide authentication data. And then discussing further with natural products, you would have to show the purity and the stability, and you also have to show how you can measure the accuracy and precision of these products. So these were some of the messages that I got.
Okay. Chris, you and I had a discussion by phone last week, and your experience was pretty deep with regard to this. Chris?
Yeah, well, I think that really a lot of the points for me have been addressed here, though. I mean, we have, synthetic reagent stability concerns. I really think the main thing I would recommend is to make sure that you incorporate regular testing into your plan. The NIH specifically is asking for regularly authenticated materials or key resources. So any kind of thing you synthesize biologically or chemically or cell lines, regularly test them over time. Just have a plan for that. I really didn't, even though I am in the instrument and systems development panels, I really didn't see anything on equipment calibration or any discussion about that being a problem. And the only thing else that I would add is that really I agree with Dr. Burnell that the system where everyone's still learning, but go ahead and get out ahead of the game and make a thorough plan. That's always definitely.
So I was going to ask you before we proceed a little bit further, when you say regular, are we talking quarterly? Are you talking annually? You're talking every 6 months? Was there any guidance with regard to that, or did it vary?
I definitely don't think there was guidance given for that, but I think it depends on the case as well. Like if a cell is supposed to be differentiated a certain way, you may need to test it every couple of weeks. Or, if a reagent stability is an issue, maybe every 6 months to a year.
Okay.
I would agree. I think if it's a transformed cell line, I think testing it once or twice a year is enough. But if you're working with something like a stem cell line where differentiation and biological properties are important, I think people would expect it to be tested a little more regularly.
Okay. Dr. Gillespie?
2 short comments. First, the study sections I've been on, just like those that you've been on, have paid sort of little attention, certainly not intense interest in the R2T issue. And so there was a lot of variation across groups. Secondly, even though it doesn't get a lot of attention, and even though not all of these criteria enter into the score, from a grantsmanship point of view, we probably ought to pay attention to the fact that doing a really good and sometimes creative job addressing these issues might buy you some points, some recognition with reviewers. So having said that, the ways you authenticate reagents are probably going to be dictated in part by the reagent. And indeed, even commercially available antibodies will have different specificities depending upon the tissues that you apply them to and other aspects of the experiment. So I think that the more us as applicants can customize not only the authentication but the other aspects of R2T in our write-ups, the better chance we have of gaining traction as credible with reviewers. That's what we want to accomplish.
All right. Dr. Levitan?
So the study section I'm on is very clinical, so I would say 80% of the grants that we reviewed either had no analytes tested or were using clinically or commercially available tests. So the attention was, did this need a plan? No. Okay, move on. For the remainder, and none of these were grants that I reviewed. It was basically, did it need a plan? Did it have a plan? So really, it was a very low bar of having that plan. But I think that's largely a function of the kind of studies.
It's a function of study section. Dr. Zhang?
Yeah, so the study section I served in, more than 50% of the grants did not have authentication, but it was just administrative notes saying this grant didn't have one. A few of the grants have really good authentications because they're using a natural compound that they're trying to demonstrate this chemical is synthesized or purified to some kind of standard. So that was commented on too, saying this grant has good plan.
All right. At this point, I would like to ask if there are questions from the audience or from anyone that is online with specifically to any of the panelists with regard to authentication and strategies for authentication of biological and chemical resources. Yes.
So what sort of things did you guys see in terms of authentication of antibodies that might be commercially?
So I've seen people put catalog and vendor number on it. It's also true, like if you submit a paper now, they not only ask for you, the company you buy it from, but also the catalog number, because sometimes one company has multiple antibodies. So people do put antibody exact number on it and chemical from which company. Well, it's a check on. No, I didn't see people showing the it detects or not. They do show where it's from. So if there's a reviewer who is familiar with that antibody and know it doesn't work, they will comment on it.
And although this isn't relevant to study section, but this comes up frequently on the journal that I'm an associate editor for, but we're increasingly requiring people to show the complete Western blot and also to eliminate the specific band with by adding the antigen, repleting the antigen into the mix to show specificity of the antibody. So even with commercial antibodies, that kind of rigor, this falls probably better under scientific rigor, but even with the commercial antibodies, let's show the whole blot, and you take the specific band out by blinding the antibody with the specific substrate.
Yeah, so I've seen people saying the authentication of the antibody will be demonstrated using a blocking peptide or using an alkyl mice. So there are people doing that description.
Yeah, so I've been on a couple of study sections that have done this as well, and from my experience, antibodies were probably the biggest thing that people picked on. A lot of the grants were, yes, they have an authentication section that looks fine, just like what we do with the animal subjects or the human subjects. If everything's there, it's fine. They don't even discuss it. But there were a lot of people that occasionally a few grants really picked on antibodies, especially with issues with Santa Cruz antibodies and their future being non-existent after whenever it is. But anyway, and so just saying that we're going to use this lot is not good enough, at least from the experience that I had. You've got to show that it really is, because you may have, and you know, I go through this all the time with antibodies to one protein. We've looked at 5 different antibodies, and they all give you a different result, but yet they're all sold as the same thing.
So also, if the preliminary results show Western blot that's not really up to standard, people will use that to comment, criticize.
I think in terms of antibody, I think it really depends on what a target you work on. If your target is, the antibody is critical for the grant application, for example, for diagnosis, for therapeutic purpose, of course you have to authenticate this antibody. That it's been routinely utilized by a lot of lab and commercially available. Of course, Santa Cruz is 50%. But for those antibody, it will be called less attention by the reviewer. So I would think the antibody specifically for the target that you work on, and particularly when you try to use it as a marker, biomarker, or diagnosis reagent, those key reagent you have to be a very normal antibody. But if you focus too much on the antibody, this grant will be going nowhere to be novel, because it will just talk about antibody.
All right, and because we do need to manage the clock and be mindful of people's time, I would like to move on to the other one that has drawn a fair amount of attention, and I will again go through the order with Tulane and then to Auburn and USA. How much discussion and how much, or with regard to strategies and attention, was paid with regard to consideration of relevant biological variables, especially in certain studies such as focusing on sex or age or specifically other biological variables? How much attention was drawn to that, and was it just an acknowledgement of, yes, we're going to include both sexes, or was there more discussion than that?
So on the panel that I sit on, this first time that we did it, there actually was a reasonable amount of focus on this. You couldn't just pay it lip service in your application. They wanted to see scientific justification and scientific rationale. For example, if you wrote a section that said, well, we've always used female mice for these studies, and we're going to continue to use female mice because all of our data previously has been in female mice, that was really unacceptable. You really had to have a scientific rationale, justification for why you might only use female mice. So, for example, you work on ovarian cancer or you work on breast cancer. Okay, we understand it. For X-linked diseases for male mice, but they really did devote a fair amount of attention to this in the first go-round. How much attention will be paid to it in the future, I don't know, whether it'll get more or less. I think this is probably one of those topics where if you work in the same area over and over and over again and you keep writing grants on the same topic, eventually you'll evolve a standard language section that you just insert in there, and then you just adjust the numbers of males and female mice. But we did pay a fair amount of attention to it this time.
Dr. Liu?
Yes, I agree to what Bruce just mentioned. And of sex, and I think it really depends on what you work on. And so obviously to diabetes, a lot of these diseases and that associated with both sex. So you have to pay attention. With the specific area, specific male, female type of disease, you don't need to worry about that. And in terms of the variation of the biosystem, and I think if you work on the yeast system, continue to keep work on yeast system and try to get a grant from NCI, it's very difficult. And if you try to use, I see a grant that uses zebrafish to try to screen a drug, and a Drosophila model to screen a small molecule drug, and that is probably challenging. So I know a lot of people try to emphasize in terms of a cancer-related research, because I've been serving a study section at NCI-related research. So they started to emphasize using PDX, so patient-derived xenograft model. So if you're missing that, they will criticize you as well. But because I'm not reviewing other type of cardio, diabetes, neuroscience, and developmental biology, so that part I cannot tell too much.
Can I add one thing?
Sure.
Can I add one thing? So one thing I was going to mention and completely forgot is that we had a few people that tried to say, well, I've used male mice in the past. You're asking me to add female mice. I can't do that because the budget won't allow it. That went over very, very poorly. So that's one thing to tell people is don't use budget as a limiting factor for doing these dual gender studies or dual sex studies.
All right. Vishnu?
Yes, it's interesting. The last comment, I've seen it in the last few panels, and also since you have to explain or justify, now they are trying to use the word-word section to justify the biological variable, sometimes exceeded more than 2 pages. So that didn't go well with
You gotta have it. All right. Chris?
Yeah, I think, again, I have little to add to the discussion. We have really the main thing from my panels that caused problems were, again, using only male mice, only female for certain projects where it wasn't warranted. And really, I don't know how often this happened with the other panelists, but there are many, still many grants, and some that I'm reviewing now on the special emphasis panel that this wasn't really addressed. It should have been addressed and is not.
And it is a scoreable criteria. So, Dr. Gillespie.
Right.
I think it's worth reiterating the need for thoughtful justification. I did a P50 review not long ago whose focus was on trauma, and they argued that they were going to study male animals because the vast majority of trauma occurs in males. And they said that they would come back with ancillary studies and examine female populations, and nobody balked at that, maybe because they didn't really think about it or care about it. But the take-home message for me was that you thoughtfully justify your system relative to gender. Most reviewers will cut you some slack on that. It's interesting, too, that cost, we all know cost is a factor, and yet us as reviewers won't accept it. So it gets back to justifying what you do. Thanks.
Yeah. And the NIH people wouldn't accept it either when we in our study section.
I have a question for Mark. It's okay. Did they show statistics of males being affected by trauma? Did they give any citation?
You know, I can't remember. We paid so little attention to the matter that it came and went in a single sentence. Maybe next time. They didn't get funded, so the next time they might.
I think for in terms of model system, if in your grant you have a cohesive, you can cohesively utilize a multiple model system, it will be benefit to your study. Of course, it needs to be relevant.
Right. Dr. Levitan?
So a lot of the grants that I review are ancillary studies to previously established cohorts, many of which were actually established as cohorts of men or cohorts of women.
Oh.
And so what a lot of people have done is go in with multiple cohorts. That brings in its own set of problems because then you have to essentially harmonize variables across cohorts, and that was not actually particularly stressed, but that was used as a strategy to address the sex as a biological variable.
The reason I'm looking at it that way is I don't want to be the guy who has to reconcile all those variables.
My guess is that if they actually get the money, they'll do it in parallel and not together. Right. But I don't know. That was not.
Dr. Zhang?
So in my study section, every grant was commented on whether there is a justification of sex, but it wasn't commented on prolonged discussion. It's just whether it has it or it hasn't. Also, in neurodegenerative disease, every reviewers know there's some sex bias on Parkinson's and Alzheimer's, and they're just looking for justification.
Okay. All right. So moving ahead, well, are there any questions from the audience or anyone that is online with regard to the consideration of relevant biological variables? Was the whole focus on sex? Was there any other biological variables brought up, or in general was it focused primarily on sex? I'll just throw that out to the whole group from me. Anybody can
I mean,
answer.
my study section was primarily sex focused.
All right.
So there were questions about age on my study section, but it wasn't really brought up as part of this R2T thing, just as more as a question.
Sorry. Thank you. So coming back to that, the age and even the sex, I just want to make sure, I'm just starting to write, is it if there may be variable, it may be important in the disease that you study, but if you are not focusing the research on establishing a sex effect, what we need to do is just make sure that the study includes both sexes, or does it need to be necessarily matched? Or how far do you need to go?
If you, if this is a question, right?
Yes,
Are you asking a question?
How far do you need to go? You just need to make sure that the study will include both sexes, or does it have to control for possible sex effects?
I think as long as it's not sex-specific relevant question, it's a general question, you better include in equal number of group mice or rat, equal number of group sex into the studies to the research experimental group, and that's all you need to say. However, if there are phenomena like literature that show some sex bias, you may want to address that, because some reviewer may catch that issue and then say, oh, so you want to pay a little attention if you use both sex group. But you want to say, for example, you do a control and drug or whatever experiment or genetic knockout, and you want to see, I will include in both male or female group. I think that's all you need to do.
Okay. Yeah, it's
the one thing that I would add,
David, because this is a statistical question, is it not?
Yes.
If you have a group of 12 animals, 6 of each gender, how do you frame the question statistically? David, are you still there, or did you go get a beer?
I went and got a beer. No, I'm still here, and I'm going to answer the question based on the advice I've been giving everyone else that's been coming to me, because I have a lot of animal researchers who are saying there's just, and this goes back to Dr. Gillespie's comment about there's just no way this is going to bust the budget. This is going to blow my budget out of the water. And I keep emphasizing the mandate is not that you must power to detect a sex difference. What they're asking you to do is to recognize that sex, in a sense, is an environment, and it is a factor that could very well affect your research, and that you want to have that, you want to see that variation, you want to study that diversity, and you would like to, you asked the question, I think the phrase that you would, should we control for it? The answer is yes, but it's one thing to control for it, and it's a very different thing to design a study to power it to detect a difference. I have not seen a mandate from NIH to say, you've got to detect a sex difference. It may not be relevant. What they want is for you to capture the diversity and observe the variation and hopefully have more generalizable results.
That's fine. And
it's a tough situation for us all, because if there are unknown gender differences in your group of 8 or 12 animals, you don't get I'm painting a hypothetical situation, but we've all encountered it before. If there are gender differences and you do lump both genders into your group and you don't get a significant difference between them and control, then where are you left? You can plot a scatter diagram that points to the prospect that a gender difference between the treatment animals and the control group was responsible for the inability to detect a significant difference, but you don't reach P.05, and you don't get refunded, and you don't publish a paper. Now there's a rosy picture.
No, I'll get that paper published. No.
That's why I like
I think this is going to be an endless debate if we talk about that detail. But within the 12-page, you just try to address what need to be addressed. Like David mentioned, if sex is not issue, then why you need to address it? Because in the clinical point of view, no, for any medicine we taken it, for a headache, there's no direction say male take 2 pill, female take 1 pill. It's only an age difference. So therefore, in most of the clinical case, except the sex-associated disease or a phenomenon, otherwise we more or less similar, except the hormone-derived event.
Dr. Liu, I agree this is an argument without, or discussion without end. But since this whole topic of R2T is evolving, there's an opportunity here to lead the way on the argument. And
Sure.
One way is to say that I'm not interested in sex-related differences, and because I'm not interested in that, I will accordingly study the same gender in control and treatment groups, and it will be male or it will be female. The other way is to
as long as you address that. I agree. As long as you address that, and then I think everyone, because reviewer are among of us. We are the reviewer, we are the applicant as well. So we all understand the same situation. We are not reviewing alien, and those review are not alien who review ours. So we are on the same page, I believe.
All right, for the sake of time, because we do want to try to keep it and finalize about 6. My next question is with regard to, and if you've got strong opinions, I'll invite you to speak up. With regard to scientific premise, how much discussion was given to scientific premise? Was it still treated very much like how thorough was the scientific literature review, or was it much more along the lines of being, let's say, a critique or an evaluation of the published literature? Dr.
So
Burnell?
for our study section, we actually like this change if people handle it in an appropriate way. I mean, I still think the NIH really doesn't know what they want it to be, but some of the better ones in our study section, it wasn't just, oh, we all read these grants, and I'm not going to pick on any single disease, but lysosomal storage diseases are bad. They kill a lot of people. I have to study them. The more effective ones in the study section that I was on were handled where, this disease is horrible. It kills a lot of people. I'm going to look at this aspect of it, and that's important because, and that's going to shift the way this disease is understood because, and adding that additional part of it really allowed the reviewer to say, all right, I agree or I disagree, because it was clearly stated, and it was clearly evident that you had thought about what you were doing, why it was important, and more importantly, how it was potentially going to change our understanding of disease or the way that we treat a disease or the way that we do drug development. That was very important in our study section.
All right. Dr. Liu?
I'm sorry.
No, that's all right. Go ahead.
Can you hear me?
Yeah, we can hear you now.
Okay. So to me, there's actually not so much difference, except you just mentioned the premise, because now they want to identify the weakness, the gap in this area that you try to investigate, put money on.
Correct.
Instead of just strengths. So the topic. So I think to me, except you change the term, nothing dramatic difference than used to be.
Chris and Vishnu, do you both?
The conclusion is that you still want to propose research that have a strong significance in the field and high impact in the area you work on, and also identify so-called weakness, so-called gap. Gap means something that still needs to be addressed. Then, to me, there's no difference between now and then, except they change the term. However, when you put the proposal, I do put premise into the significance page and also the innovation. I think the significance page as well as the background page, introduction page as well. So that's all. I just changed the term in terms of the impact and also the gap. It's no different to me. That's all I want to say.
Okay, thank you.
I would like to state something. Hello?
We've got you.
Yeah, it was well stated by, I don't know if some of you have read this, but Mike Law, one of the assistant directors of external programs at NIH, specifically mentioned a good example. If enzyme A affect a specific function of a cell, that's the proposed plan. And then the preliminary data shows that enzyme A modifies protein B. Existing literature shows that protein B alters the specific function of the cell. Now the entire strength of the proposal is dependent on the existing literature. So now you have to critique, you have to look at the weaknesses and strength of the existing literature. So this has to be clearly stated. This was well stated by Mike, one of his articles. So I thought I should share that with you. So.
Chris?
Yeah, I think I would definitely, some great points were raised. I would just say that this is an important criteria. It's significance really is almost the same thing, just a different way of saying it, but they are making the reviewers specifically say the word premise. What is the scientific premise? So why not just help the reviewers help you and put it in there? The premise of my work is this, right?
In other words,
yeah, some of the most effective ones, and most of the effective ones in our study section, they actually delineated a specific section to scientific premise, where they summarized everything.
Okay. Dr. Gillespie?
This is quick. Nothing to add.
like a good study section
This
member.
is the, yeah, nothing to add. I thought this was sort of an unusual criteria to add because it basically says exactly what the old PHS 394 forms did years ago. Articulate the gap. It makes it easier for reviewers, I guess.
Okay. Well, Emily or Dr. Zhang?
So the SRO basically said that the study sections had been evaluating both premise and rigor, but because the NIH was watching, that we should use those words in our reviews.
Okay. So in other words, and this is something that I think I want to echo before we go a little bit further. I'm not going to forget. This is something that we've said over, going all the way back to the very first forum in December. These really aren't new. They're asking, especially for scientific premise and scientific rigor, they're just asking for a flashlight and a highlight on how you're going about and what is the premise of this and how rigorous are you in the design of this experiment. Dr. Zhang, what's your experience, please?
So my impression is in the past, for example, Parkinson's disease research, if the investigator is saying Parkinson's disease is bad, alpha-synuclein is involved, then it's already significant if you're dealing with those two issues. But now the reviewers look a little deeper, like what exactly, which aspect of Parkinson's disease you're studying. So in the past, the critiques are in the research strategy portion, and now they move it to the significance portion because they will comment on whether you are biased and really wish your approach will work, or you are aware there are other complications.
All right. Any questions from the audience or anyone that is online? All right. In the sake of, we've got about 6 minutes, so I'm going to, instead of going around the room, around the horn again one more time, I'm going to bring up, we've got a lot of emphasis on scientific rigor and a lot of resources online at this time with videos on the CCTS webpage indicating discussion of each of these. I'm just going to throw out to the panelists, if you have a very strong comment or a thought with regard to scientific rigor, have the approaches changed? Not so much has the attention changed, but have you seen a change in the approaches to it in response to these, the new mandates and the new instructions that have gone to study sections? Has anybody seen anything that has changed with regard to the designs or the approaches statistically, or more emphasis on statistical power? And I'll start with Dr. Bunnell.
So, I mean, I think, you know, in the field that I work in, if you were writing an effective grant, you were doing power analyses for your animal studies already. So in my world, this really didn't change much of anything because you were already doing it.
All right. Dr. Luke?
Yeah, I don't see any change because the last study section is the first study section right after this change. I would, you know, anticipate see maybe a little bit more dramatic change a few years later after a few run, NIH keeps emphasize these things. And in terms of, let me see, in terms of the unbiased, I would recommend any of you, we us, including myself, sitting here to emphasize any of your in vitro experiment performed by two independent pair of hands rather than just one person, or can be performed by someone else. Besides, of course, propose animal study when you have more power, enough animal. But all in the animals work, try to mention you doing the blinded work. Like you know, let one dissect animal, label the number, another to analyze the data without knowing which is which. So emphasize those. This will maybe address those unbiased outcome or experiment approach.
That is something
that's what I do in my grant. Yeah,
that's
what I recommend.
That's what we've been recommending here at UAB, is not to emphasize just purely statistical or analysis plan, but to emphasize design, and in some manners to be able to blind some of the information to allow for greater generalizability, as well as letting an independent person be able to look at the data and see if they can replicate the information in a blinded manner. Vishnu or Chris, your thoughts?
Yeah, I've seen a couple of those examples in the last round, two collaborating labs doing the same experiments to assure reproducibility, and certain samples were sent to both certified pathologists for immunohistochemistry. They were non-sequential and blinded samples. Collaborating labs developing SOPs for consistency and cross-training of graduate students in specific techniques.
All right. Chris?
Yeah, my view on it, again, I think it is very similar, again, to what, you know, a good research design, we should have been doing this anyway. Plans, backup plans, milestones, things like that should be included. It's kind of an old review criteria. They are making the reviewers say, was it rigorous? Using the word is kind of awkward, actually, in trying to figure out the best way to use that word, for me at least. What I would say is sort of the opposite of what I said with premise. I'm hoping this doesn't kind of suggest that people should use the word rigorous, like we're going to do our science in a rigorous way. It's kind of an empty type comment. I'm hoping that it doesn't go down that direction. Just basically continue to develop well-designed plans.
All right. Mark?
Nothing to add.
Thank you, Mark. Thank you very much. Dr. Zhang? I'll go with you first, and then I'll go Dr. Levitan.
So I think the rigor is always important, especially if the differences you're observing is really tiny. That will be commented on. If the data is not robust, it will raise a lot of eyebrows. But our study section also discussed about we're still reviewing for impact, not reviewing for just, it's not an approach-based grant. It's still an impact-based grant review. So I think people sometimes overlook how much statistics and power analysis, whether even power analysis is there, if the data is like really yes or no, they don't really ding the applicants on it. It's a combined issue of impact and rigor.
Okay. All right. Dr. Levitan?
The only change was in language, not in
the content. You're kind of like Chris. You started saying the word rigorous, but you didn't see necessarily the change. Okay.
Well, it's always been, I mean, it's always been an
Well,
important criteria.
Well, you see, this is one of the things that, again, it's a greater emphasis. It's not that it had been lacking. I've been on study section for 10 years for the stated purpose, evaluate the design. But the one thing I would add is not just to think about it as what is an analysis plan, much more of how do you design it to, as Dr. Liu was pointing out, to try to reduce the potential confounding due to one individual having too much influence within one experiment. Can you divide responsibilities? Can you blind the samples so that you do not bias people toward one interpretation or one conclusion? And these are all in the resources that we've placed online. Given that we are now at 6:01, I would like to be able to thank all our panelists. I will take, if there is one more question from the audience or from anyone online, I'll be happy to take it with regard to scientific rigor. But if not, I would like to thank all the panelists for their time and for allowing me to talk to them last week and tolerating all my emails over the past 2 weeks. And I do greatly appreciate your time, and I think this has been very, very successful. Thank you very, very much.
Thank you, Dave.
Thank you. Happy to do it.
Sorry for not able to call you.